Good afternoon, everyone, and welcome to today's ESNM GMFH webinar. I am Joël Doré. I'm Research Director with INRAE in France, member of the Board of Directors of Gut Microbiota for Health since its inception in 2012, so just 10 years ago. I'm honored to chair this today's session of the ESNM GMFH webinar series on Clostridioides difficile. Today we are together for 1 hour for a session that concerns pathogenesis of Clostridium difficile, and we will start with 2 presentations and end with a question and answer discussion. The first presentation will be given by Ben Molish on C. difficile as a model of dysbiosis, the role of microbiota and microbial metabolites, and the second presentation will be given by Ed Kuipers from Leiden in the Netherlands on overview of Clostridioides difficile infection pathogenesis. Now, before we start, I would like to acknowledge the support from Ferring Pharmaceuticals for the ESNM GMFH webinar series 2022. And also we'll start with the webinar housekeeping aspects, just to give you attendees clues as to how we can deal with the question and answers. So you will have the opportunities to submit text questions using the Q&A box that you will see at the bottom of the control panel, bottom of your computer or your iPhone. And we prefer that you do use the Q&A feature that provides an opportunity for you to enter your scientific questions for the panelists to respond at the end of the webinar. And you can do this all along the webinar. We prefer this to the chat button that may cause some difficulties, so please prefer the Q&A. And then just press send when you have written your question, and that will be addressed at the end during the discussion session. Interaction is important to us, so please do not hesitate to send us your questions. I mentioned already for the chat, and we have an option for a pause, but we will not be using this option, I think, today. One last remark before we get started: the webinar will be recorded. The recording of this webinar will be available at a later stage on the ESNM GMFH YouTube panel. And let me introduce our first speaker. After I quit sharing, okay. Ben Molish has a background in gastroenterology and hepatology, started the FMT service at Imperial College London back in 2014, and was first author of the UK FMT guidelines and contributed to a number of other European guidelines on the subject. Dr. Molish then undertook a PhD at Imperial College London, with supervision by Julian Marchesi, looking at microbiota metabolome interactions in CDI and the impact of FMT upon this. He's now a clinical lecturer at Imperial, further investigating microbiota metabolome interactions to explore mechanisms of FMT in a variety of gut and liver diseases conditions. Ben, if you want to put your first slide, the webinar is yours.
Thank you very much. Hopefully people can see my slides now.
Yeah, we have the presenter mode.
Great. Okay, here we go.
All right, perfect. Thank you.
Thank you very much. Well, thank you very much, the ESNM, for giving me the opportunity to present to you today. Just to give you a few slides of overview, an introduction to start off with. For several decades now, we've had this concept related to C. difficile infection of colonization resistance, and that C. difficile infection is a clear example of the importance of the gut microbiota in colonization resistance. In other words, there's intrinsic properties of the gut microbiota to protect us against enteric and other infections. But if you go back to even 13 years, this was the sort of paradigm that was being used. In other words, we recognize that broad spectrum antibiotics are a key risk factor for CDI, and that the more that someone is exposed to broad spectrum antibiotics, the more the disruption of the gut microbiota, the greater the CDI risk, and that this is abrogated by removal of antibiotics, and so this risk removes itself. However, it was only really over the past decade, a little more, with the arrival of next generation sequencing, the explosion of next generation sequencing, that we began to unpick this in any great detail and exactly what the gut microbiota contribution to colonization resistance is. And here we go back from just over a decade ago, when it started to recognize that the more antibiotics are given, the greater the disruption to the diversity and the taxonomy of the gut microbiota, and the greater the risk of C. difficile infection. That was the paradigm approximately a decade ago. To give a little introduction to microbial metabolites, which should be a key part of this talk, gut microbial metabolites are a key intermediary between the gut microbiota and the host, sitting at the interface between the two, and being one of the key mediators of the effects of the gut microbiota on host organ systems, immune system, and other aspects of host physiology. What do we mean by gut microbial metabolites? Where do they come from? Some of these are more on the left-hand side of the figure, in other words, related to dietary components, and particularly short-chain fatty acids, which represent fermentation of undigested dietary components. We will also make reference to things that are more on the right-hand side, which are metabolites that are produced by the host, but are acted upon with biotransformations occurring through the actions of the gut microbiota. Another key aspect of this talk is going to be related to fecal microbiota transplants, FMT. And of course, it was 2013, this will be familiar to many in the audience, when the first randomized study was published on the use of FMT, stopped at an early stage by an ethics panel because the patients with recurrent CDI receiving FMT did so much better than receiving standard of care with vancomycin. Even in that original paper, there was some investigation of gut microbiota composition pre and post FMT, and this sort of key figure whereby there is an increase in gut microbial diversity after successful FMT to a level comparable with donors. So not only does FMT an effective treatment for recurrent CDI, but it restores gut microbial diversity and taxonomic composition. And as such, it was recognized that this could be a tool to better understand colonization resistance, at least to it relates to CDI. A further intriguing study in this area was a small study reported by a German group a few years later in gastroenterology, showing that sterile filtered FMT, in other words, filtered through a very narrow pore filter beyond narrower than the typical diameter of bacteria, could have comparable efficacy in treating recurrent CDI to conventional FMT, raising the intriguing idea that there could be soluble mediators of efficacy rather than just leaving live bacteria alone. What could those mediators be? Options might include bacteriophages, the viruses that live on bacteria, bacteriocins, so they typically proteins produced by bacteria which have an antibiotic effect, or could this be bacterial components such as enzymes or bacterial metabolites? So having introduced those sort of key concepts, I hope to go through in a bit more detail some of the most, where there's been some of the most work related to microbial metabolite interactions as it relates to C. difficile infection. And these are the sort of topics that I will be introducing. Short-chain fatty acids are a key area of interest. So just to give an overview of these are volatile metabolites that are produced by the gut microbiota through fermentation, principally of undigested carbohydrates, but also through some protein components as well. And as is shown on the figure here, these have a variety of overall beneficial effects as regards the host. They have beneficial effects regards the gut barrier function, different effects regarding immune regulation, effects regarding gut endocrine function. And as can be seen towards the bottom side of this figure, even epigenetic effects related to effects on aryl carbon receptors, as well as other receptor systems, with a net anti-inflammatory pro-health effect. As regards to C. difficile infection, there's been some work going back for over a decade, recognizing that short-chain fatty acids produced by the commensal bacteria of the gut microbiota can suppress the vegetative growth of C. difficile and inhibit toxin production. What about in the context of FMT? What happens to short-chain fatty acids? This is some work that we published looking at changes in short-chain fatty acids, including acetate, propionate, butyrate, and valerate between pre and post FMT. And as you can be seen, even from week 1 afterwards, goes on to week 12, there's restoration of these fecal short-chain fatty acids to levels comparable to that of donors from the almost negligible levels found pre-FMT. However, how do you interpret this data? It's difficult because does that mean that people are just changing their diet? Could they be eating something different because they're feeling better and the diarrhea stopped? Or could that be related to restoration of gut microbiota with the ability to make short-chain fatty acids? One intriguing study, even if I say so myself, that I was involved with is trying to take out some of those confounders by using a chemostat system. And this was principally led by my colleague, Dr. Julie McDonald. And she used this chemostat system or robogut system, an in vitro gut model, to mitigate some of these confounders. For instance, the nutrient feed that's placed into this model of the distal gut is pretty stable. So you're accounting for the effect of diet as one obvious confounder. What she found is that she induced C. difficile infection by treating her chemostat with antibiotics, exposing it to C. diff spores. And then she treated one set of vessels with FMT and one set of vessels with a saline control. And what she found is that while certain short-chain fatty acids recovered even prior to the FMT, namely butyrate and propionate, the valerate only recovered with an FMT and didn't recover in those treated with a saline control. When she looked further at valerate, which is the 5-carbon amino acid, she found this almost dose dependence between how much valerate this was some further work she did in vitro, how much valerate she could incubate with a variety of C. difficile ribotypes, and a reduction in C. difficile vegetative growth. Valerate seemed to have this very specific effect on the growth of C. difficile because it didn't seem to have any effect or any modest effect at very high doses on the growth of other gut commensals. In a mouse model where mice were either treated with glycerol trivalerate, she used glycerol trivalerate instead of valerate because of the volatile nature of it. So she used glycerol trivalerate that was liberated by lipases in the gut. She gave mice either glycerol trivalerate or just PBS alone, and found the mice treated with valerate had this big drop-off in the CFU count of C. difficile infection. What about other organic acids? So one of particular interest has been succinate, an organic acid produced by fermentation by commensal bacteria and degraded by the commensals to propionate, which is used for an energy source. After antibiotic treatment of the gut microbiota, there can be seen this big drop-off of acetate, butyrate, and other short-chain fatty acids, but this big jump in the level of succinate in the gut. And that's because in the absence of commensals that utilize this as an energy source, there's less competition for the succinate. However, C. difficile has the ability to exploit this as an energy source and to synthesize butyrate from the succinate, which can use as an energy source itself. Again, referring back to FMT as a tool to investigate colonization resistance, recent work has demonstrated that FMT can respond either with a healthy human microbiota or since a mouse work where succinate-consuming bacteria were used alone, that's the Phascolactobacterium over here, reduced the availability of luminal succinate and outcompeted C. difficile for the source of succinate. And this will be a theme that we'll return to several times, the ability of C. difficile to exploit carbon sources for its growth in the absence of commensals, but flooded out when commensals are restored by something like FMT. Moving on from short-chain fatty acids to another metabolite group, amino acids, C. difficile has this unusual property in that it has the ability to use the Stickland pathway to exploit amino acids as a sole energy source. The Stickland pathway shown over on the right here is a joint oxidation and redox reaction of a particular amino acids. Amino acids also have this slightly contradictory effect whereby they can reduce toxin yields of certain C. difficile strains as well. Going back to this idea of what happens in the context of FMT, this is some work from the Mayo Clinic published several years ago, in which dysbiotic mice, so in other words, mice with a disrupted gut microbiota, were found to have a lot of proline in their gut and to be able to, and that C. difficile was able to exploit that as an energy source for its vegetative growth. After FMT into these mice, C. difficile was outcompeted for its use of proline, couldn't use it as a Stickland pathway substrate, and the growth of C. difficile was reduced. Another intriguing amino acid, but relevant in a slightly different way, is related to tyrosine. So C. difficile also has an unusual property in that it can ferment tyrosine to this chemical called P-cresol via an intermediary P-HPA, as shown in the figure on the right here. P-cresol was thought of as pretty toxic to lots of bacteria, particularly gram-negative bacteria, because of its bacteriostatic nature. However, unusually, C. difficile seems to be somewhat resistant to the toxic effects of this. So C. difficile can produce this particular metabolite. It can survive. It doesn't cause any toxic effects to it itself, but it can use it to kill off other bacteria, particularly gram-negative bacteria all around it. Again, using outcompeting other bacteria that fill the same ecological niche. A number of different sugars are of key relevance to C. difficile. There's an intriguing story from a couple of years ago regarding trehalose, of which you're going to hear a little bit more in the second talk. And some recent work from Justin Sonnenberg's lab in Stanford has related to sorbitol use. Justin Sonnenberg's lab looked at what was different about what happened in the inflamed gut. So they took a mouse model of C. difficile, and they used C. difficile that made toxin and compared to C. difficile that didn't make toxin. And their hypothesis was that the inflamed gut that occurs in C. difficile with toxin might have availability of metabolites that it can exploit to use for its growth. So they compared, using RNA sequencing, the gene profiles of transcriptional profiles of the gut of a C. difficile toxin-producing mice and a C. difficile non-toxin-producing mice. And they saw this overrepresentation of sorbitol-related genes in the toxin-producing mouse, and found that sorbitol was able to be used by C. difficile as a source of energy. To move on to the last group of metabolites, but perhaps those that have been best studied, I want to talk a little bit about the work that's been done on bile acids. Bile acids are a sort of co-metabolite system as relates to bacteria. These are produced as cholesterol derivatives by the liver, secreted through the biliary tree into the gut, and when they're secreted, they're conjugated to amino acids, either taurine or glycine. They go through a variety of biotransformations within the gut, which occur through via gut bacteria. So these are a number of different enzyme systems that are present in gut bacteria, but aren't present in mammals. Of particular note are an enzyme system called bile salt hydrolases, which remove the taurine and glycine group from these bile acids, and a particular group of enzymes called 7-alpha dehydroxylase, which convert so-called primary bile acids to so-called secondary bile acids over at the bottom of the figure here. What's the relevance of these? Well, in simplest terms, as you can see towards the top half of the screen, these so-called primary bile acids tend to have a pro-germinant effect for C. difficile, shown from initial in vitro studies and later in vivo studies, while these secondary bile acids have a key anti-C. difficile effect, inhibiting its growth and preventing toxin activities. So one theory that's been around for several years is, in the antibiotic-treated gut, where there's been a loss of commensals, do we remove bacteria that produce these organ systems, have an enrichment in these bile acids at the top, the primary bile acids that are pro-C. diff? Do we lose the secondary bile acids at the bottom that are anti-C. diff? And could that contribute to CDI pathogenesis? Conversely, could FMT restore bacteria that produce these enzymes, and so restore the normal bile acid milieu in the gut? And the short answer is yes, there's a lot of evidence to support this. So this was work from Jessica Allegretti in Harvard several years ago, comparing patients who were controls, those with primary C. difficile infection, and those with recurrent C. difficile infection, and looked at the level of predicted BSH abundance in the guts, and found this was reduced in patients with recurrent C. difficile compared to groups. This is work from Eric Paymer's lab from Nature several years ago. They used mathematical modeling on human samples to try and investigate which bacteria provided colonization resistance against C. difficile, and found their top hit was were bacteria which had the ability to bioconvert primary to secondary bile acids, particularly this bacteria here, C. scindens. They showed in a series of experiments that either a 4-bacteria combination or just C. scindens alone, which has the ability to bioconvert primary to secondary bile acids, was able to protect against C. difficile infection. We have some work we've performed with Jessica Allegretti and colleagues, looking at people with primary C. difficile infection who go on to recur versus those who don't go on to recur, and looked for changes in the bile acid dynamics. And we found that patients, the short story of this is we found that patients who don't recur have a greater recovery in secondary bile acids and have a greater recovery in BSH across several weeks compared to patients who go on to recur. While I talked about BSH and 7-alpha dehydroxylase in particular, there are actually a variety of microbial enzyme systems in the gut that bioconvert bile acids. This was some intriguing work last year that looked at centenarians and their bile acid profile. The hypothesis being that centenarians, having survived to such an old age, might have some aspect to their gut microbiome in terms of its functionality that might have protected them against infections and other trials and tribulations over the years. And they found that the centenarian gut microbiome is enriched in this particular isoallolithicolic acid. And then on further examination found that this isoallolithicolic acid had a potent specific activity against a number of pathobionts, including C. difficile infection in particular. What about going back to this FMT or therapeutic aspect? We were involved in some work showing that FMT restores gut bile metabolizing enzymes, and particularly BSHs, which accompanied a reduction in taurocolic acid. There's been a number of interesting next generation FMT products. This is a spore-related product produced by Series Therapeutics in the USA, was shown in a recent randomized controlled trial to be effective as a treatment for C. difficile infection as an alternative to conventional FMT, and was accompanied by restoration of secondary bile acids. Newer proposed anti-C. difficile antibiotics that have a more specificity against C. difficile than older agents appear to be more protective of the gut microbiota and more protective of conventional bile acid profiles. And there's been some suggestion that interruption of bile acids interacting with C. difficile via the bile acid receptor of C. difficile may be another therapeutic option. These 7-alpha dehydroxylase producers may have a number of off-target effects as well. The same bacteria that produce secondary bile acids may also produce trypsin-derived antibiotics and may also have some effect of the stickling pathway as well. Bile acids don't just work, though, apparently through their effect just on the C. difficile life cycle. A number of microbially derived bile acids also seem to have potent effects to regulate our immune systems, particularly regulatory T cell function. And we were also involved in some work showing that FMT not only restores the normal bile acid milieu, but is accompanied by an upregulation of FXR activation. So it could be that bile acids are having a number of off-target effects that are such as FXR regulation that are associated with regulation of gut barrier integrity, immune response, etc. So to conclude, there is a complex interaction between the gut microbiota, gut microbial metabolites and CDI, and there's many other metabolite systems left to explore. Perhaps the benefits aren't just within the gut itself. This is some work we've been involved in looking at short-chain fatty acids in serum pre and post FMT, and adding this to some astrocytes and microglia, looking at their immune responses and showing that they can change the immune responses, perhaps hinting at there being the FMT changing the gut-brain axis by short-chain fatty acids as an intermediary. And for me, FMT appears to be really a good discovering tool for discovering metabolites contributing to colonization resistance. And this is just metabolites. My remit here is just for metabolites, but as alluded to earlier, there are a number of other mechanisms that seem to be important for FMT, including bacteriophages, bacteriocins, and others. So there's plenty still to look into here. Thank you very much for your attention, and thank you very much to all the team here at Imperial and Beyond for all their help and support with some of the research shown here. Many thanks.
Thank you very much, Ben, for this wonderful presentation on the impact and the interplay with the bacteria and metabolism. We'll keep the questions for the Q&A session at the end. And I will now introduce our next speaker, which you should see, Ed Cooper. Ed is medical microbiologist and researcher at the Laboratory of Experimental Microbiology, National Reference Laboratory for Clostridioides difficile at the Department of Medical Microbiology, Leiden University Medical Center. Dr. Cooper is advisor for the Center for Microbiota Analysis and Therapeutics, CMAT, and senior expert at National Institute of Health, RIVM, for the theme of antimicrobial resistance. He's currently chairing the study group for host and microbiota interaction of the European Society for Clinical Microbiology and Infectious Diseases. Ed, the webinar is yours now. Thank you.
Thank you very much for this nice introduction, and thank you, Benjamin, for this excellent first presentation. I have 4 topics: the changing epidemiology, the colonization and development into disease, how does C. diff spores germinate and grow out and produce toxin, and finally, what are the consequences for interventions. So probably you know this excellent review paper written by Klaus Smits from our department. So the intestinal tract can be colonized with C. diff without any symptoms, asymptomatic carriers yet. Usually the CDI is presenting as a hospital-acquired diarrhea after the use of antibiotics. The severity of the disease can vary from mild self-limiting diarrhea until cytomembranous colitis. And so we do have a clinical picture of community-acquired diarrhea. The complications are especially severe inflammation with a toxic megacolon, perforation, relapses 20%, or a reactive arthritis, which is very rare. So the problem is the high mortality. That's an overall mortality, not specifically related to the C. diff infection, but also associated with the underlying comorbidity. And the second problem is the recurrence rate in 20% of the cases. It's a huge problem, the CDI in the US. It's estimated that there's on an annual basis about 15,000 deaths due to CDI. And that's different from the European situation. We have about 6,000 deaths on an annual basis. And usually antibiotics are the predisposing factor. However, it's not only a disease of humans, it's also a disease of animals, especially of pigs. It's therefore a One Health issue. And especially the pigs, of which are more than 130 million in Europe. And especially in pigs, the disease can be fatal and can also result in a growth retard and less economic value of the pig farm. So what happened in 2004? In 2004, suddenly new C. diff ribotypes occurred. Ribotype 027 and ribotype 176. And they probably originated in Canada and Pittsburgh, and they spread throughout the world to Europe and also to Australia. And a few years later, another hypervirulent strain was found. It was the ribotype 078. This is typical of the strain that can also be found in animals, especially in pigs. And the disease of the 078 and the disease of the 027 are different. This is usually in healthcare facilities with high mortality and rapid spread, but this is usually outside the healthcare facilities, also in the community, in a younger population. And there are not much outbreaks reported to 078. So it doesn't stop. So since 2017, we have received strain from several countries and several labs who encountered new variants of C. diff. So for instance, the ribotype 036, or that is similar as the 198, or the 181. The 181 was recently found in Greece, and it turned out to be very much related to the 027, similar as all those other ribotypes. So there are a few ribotypes that can be discriminated from each other by PCR ribotyping, but are very much similar by whole genome sequencing. And if you look into a tree, phylogenetic tree, you can see that all those hypervirulent strains are located here, separate from the other strains. So PCR ribotyping is probably not the best typing method to recognize hypervirulent strains. We should go to whole genome sequencing. What was the cause for that sudden increase of 027 strains in 2003, 2004? This was a very interesting hypothesis, and it was also proven in an animal model. So there was a coincidence with the introduction of trehalose as a food additive. And in an animal model, it was clear that if you give the animal model trehalose as spores of C. diff, then the disease of the 027 has a more severe course than without the treatment of trehalose. So this was an interesting hypothesis. Trehalose is a food additive, and it was introduced in 2002, 2003. However, this hypothesis was not confirmed in another study. Another study that was published by the group from Oxford and from Leeds, and they found that this changed trehalose metabolism. Trehalose metabolism was not only specific for O27, but also for other PCR ribotypes. And what about the difference between O78 and O27? So this was a publication from the group from Nitin Kumar and Trevor Lawley. They did whole genome sequencing and they did a phylogenetic analysis based on the whole genome analysis. And they found that in the past centuries, the first clade that was found of C. diff was the clade of which O78 belongs. And the hypervirulent clade with the O27, 244, develops late. If you compare those strain clade A and clade B, you can see clear differences, and the differences are especially pronounced here. So this is the survival of viable cells in the presence of hydrogen peroxide. And the clade B strains were most susceptible to low concentration of hydrogen peroxide. Also, if you treat the host with glucose, fructose, or ribose, you can see there's a difference in the spore production and also in the sporulation. So the conclusion was that clades B with the O78-like strains were much more susceptible to disinfectants and had less sporulation in the presence of glucose or fructose. And that can probably also explain why those clade B strains are less frequently found in outbreak situations. So after 2003, 2004, when we also encountered the first hypervirulent strains in the Netherlands, we developed a National Institute of Reference Laboratory for C. diff. So this was a collaboration between the Leiden University Medical Center and the RIVM, the National Institute of Public Health. And we had a surveillance and sentinel surveillance of approximately 20-25 hospitals. So each case of a CDI is registered in that hospital. There's clinical epidemiological information collected, and the strains are sequenced and characterized. So what is so remarkable in the past 2 years is, first of all, that we were the first who found the metronidazole-resistant C. diff strains. So during the treatment of a patient with metronidazole, the treatment failed, the diarrhea persisted, and we isolated the strain that contained the plasmid, a plasmid with a gene encoding metronidazole resistance. Fortunately, this metronidazole resistance is very rare. It's below the 1%, so it's probably clinically not so relevant. When we look into the details of the last 2 years of our National Reference Laboratory, there were some interesting findings. So we have about 800 to 900 cases of CDI, but we see that the onset of CDI is started in 45% at home. So not in the hospital, but at home. That means that we have seen many patients who developed the CDI outside the hospital and who were admitted with severe CDI or with mild diarrhea and comorbidity, and subsequently the diagnosis of CDI was made. And the death contributable to CDI remained pretty constant in our Dutch situation, mainly because that hypervirulent strain, that O27 strain, is very rare nowadays in the Netherlands. So we were successful in nearly eradicating this O27 strain. On the other hand, the O78 strain, gray one, is still frequently found and is currently number 3 of all the strains found and typed in the Netherlands. So community-associated or community-acquired CDI. I mean, it's difficult. So we performed a huge study with more than 12,000 diarrheal samples submitted by a general practitioner to our laboratory for just diagnosis of Salmonella campi and other enteropathogenics, not requested for diagnostics of CDI, but we just tested all the samples for CDI. And this was a very remarkable finding that, of course, what you expect from patients with diarrhea who go to the general practitioner, number 1 is campi, number 2 is Giardia lamblia, but Salmonella is on the 3rd place together nearly with C. diff. And C. diff is higher than the frequency of Shigella and of Yersinia enteropolitica. So this was also a case control study. So we compared the characteristics of patients with community-acquired CDI to non-community acquired CDI, and we found that 1/3 of the CDI patients had not used antibiotics and was also not previously hospitalized. So that means that the general practitioner did not have CDI in a differential diagnosis because the previous hospitalization and antibiotic use was lacking. And also that of all those cases, 10 to 20% had no underlying disease. They were just healthy, normal individuals. So this is a very interesting observation, and this brought us to the idea, okay, C. diff is not a disease of the hospitals only, it is also a disease in the community, and we need more information about the cause of diarrhea in the community in young, healthy individuals without antibiotic use or previous hospitalization. And that is still something to study. So if I go to the colonization and the development of the disease, we have a hypothesis. Our hypothesis is different than what has been published nowadays. So we think that C. diff does not belong to a normal, healthy microbiota. We think that C. diff is only present when the microbiota is already disturbed. So by, for instance, by comorbidity, or by a specific diet, or by medications, or just by a heavily contaminated environment. Then the microbiota is disturbed. This can result in an increased susceptibility for colonization of C. diff, and that can subsequently go to an infection. And it can also, when it's a patient that is a carrier without any symptoms, is admitted to the hospital, that can potentially lead to spread of C. diff spores and to outbreaks that originated from a patient without any clinical symptoms. So to study this in more detail, Quentin Ducamont did a study in controls that were just healthy patients that were admitted to the hospital and were negative by culture and by PCR for C. diff. And we had a study. We had also a group of asymptomatic carriers that were also admitted to the hospital but had C. diff in their stool samples. And in those hospitals that participated in the study, we also studied all the patients that were diagnosed with CDI. So we cultured the strains and we characterized the strains, and we also looked for the composition of the microbiota. Microbiota was studied by 16S analysis, and all the strains were studied by toxin production of toxin A, toxin B, by ribotyping, and by the presence of also the binary toxin gene. So what was so surprising was that in the control groups, the diversity was higher than in the patients who were colonized with C. diff, and then the patients who had C. diff infection. And also, if you look at, if you perform a principal component analysis, weighted and unweighted, you can clearly see that the patients and the controls, the blue one are the CDC, the colonized patients. The red one are the patients with CDI, and they cluster. And the controls are the blue one. And the controls are a little bit equally distributed among all the lines, in contrast to the CDI and the CDC. So we also did a network analysis. And in network analysis, we could see that in patients with CDI, there was an abundance of Bacteroides species, right? And also an abundance of Veillonella species. And we think that the Veillonella species are mainly from the upper intestinal tract. And by the diarrhea just occurs in more abundance in the stool samples. The control patients had significantly higher presence of Eubacteria halli and Fusicanetibacter. And also in the network analysis, it turned out in our study that Fusicanetibacter was really negatively associated with CDI. So the Fusicanetibacter may have some protective properties against C. diff. The final topic of the pathogenesis is the germination and the toxin production. So I can recommend you all to read this paper that is recently published in Nature Communication by a group from South America. What they did was they did in vivo and in vitro studies, and in vivo studies they used mice. And they inoculated the mice with spores of C. diff. And the first surprise was that it is not only the colon in which the spores come and attach to the cells, it's also the ileum, it's also the small intestinal tract. And it is not only an adherence of the spores to the epithelial cells, it's also an internalization of the spores in the cells of the colon and the ileum. So here you can see it in T84 cells. So this is not in the mice, but in the cells. You can see that in the cells, the spores of C. diff, there's some protrusions of the epithelial cells. And by the process of endocytosis, those spores are, do enter the epithelial cells. So if they go further with CACO2 cells and they study the internalization of the spores in more detail, they found that fitronectin and fibronectin were important for that internalization of the spores. So in those cell lines, you can see that for the attachment of the spores, there's no difference between the control buffer, between human serum, and between the several concentrations of fibronectin and fitronectin. But for the internalization, there's no internalization in the buffer, but in the presence of NHS and of the fitronectin and fibronectin, there's clearly internalization of the spores. And they also found that in those spores, there was a specific exosporin protein, this protein, B cell A3. If this protein is absent, you can't see any binding of the spores to the FN and to the FVN. But if you have the protein intact and use the monoclonal antibodies against FN and VN, you can clearly see that those that exosporin protein attach to FN and to VN. So finally, this is also a very interesting observation that came also completely out of the blue. So they decided to do an animal experiment with nystatin. And nystatin is mainly known as an antifungal agent. And nystatin can also inhibit the caveolin-related pathways. And that means that it can disrupt an intact membrane and can stop the phagocytosis of the pathogens or of the spores. So they had a model in which they treated first the mice with an antibiotic cocktail, then subsequently with clindamycin, was challenged with spores of C. diff. And after 2 or 3 days, the first group was treated with vancomycin, and the second group was treated with vancomycin and nystatin. And this was the body weight, and there seems to be a little better outcome of the weight for the treatment with vancomycin and nystatin. But this is really the message. So the relapse rate was significantly lower in the vancomycin and nystatin treated group. And they explained it by, okay, when you inhibit this caveolin-related pathway, the spores of C. diff cannot enter the epithelial cells, and therefore there will be a lower relapse rate, which is a very interesting hypothesis. The process of the germination of the spores, I think that Ben already told you a little bit on all the different compounds, like the small chain fatty acids and bile acids, which can activate the germination of the spores. The process of itself is really interesting. So this is the dormant spore, and this is the spore that is germinating. And central in this process of, let's say, of a germinating spore is this enzyme. And this enzyme is the SLAC. And this SLAC, when it's active, it just destroys the cortex of the spores, and the GPA is released from the internal, from the intercellular room to the extracellular space, and the metabolism can here then start of the spores that are now devoted of their cortex. But how does this enzyme become activated. It becomes activated because there are free pseudoproteases. Those free pseudoproteases are CSP-C, CSP-A, and CSP-B. And CSP-B is the most important pseudoprotease. If in this complex in which normally the free proteins of pseudoproteases are in a complex with each other by unknown co-germinant factors or by bile acids, there's a dissociation of the complex. And the CSP-B becomes dissociated, acts and interacts with the SLC, and SLC becomes activated. So this is the germination of the spores and the process that is involved in the germination of the spores. Then you have the outgrowth of the strains by external factors, of which the diet is very important. And finally, you do have the toxin production, because the disease only develops when there's a toxin production. And the toxin production by the toxin genes, by transcription, is also extensively studied. So what are the factors that stimulate the vegetative cells of C. diff to produce the toxins? And that are numerous factors. And those transcription factors, they can be induced just by the nutritional environment, but also by the host environment. And these are all those factors together. You can see it's a huge number of different factors that are involved in suppression of the toxin synthesis or induction of the toxin synthesis. So what are then the consequences for the intervention? So also based on the presentation of Benjamin, it seems that the microbiota or the intestinal environment is very much important. It's important for the attachment of the spores, for the internalization of the spores, for the germination of the spores, the outgrowth of the cells, and the production of the toxins. So all those interactions are with flora that have an influence on the hydrolysis of primary bile acids to secondary bile acids, on the microbial flora that produces the small chain fatty acids and that releases the sialic acid from the bound compounds. And it should also overcome the factors that inhibit the C. diff growth, like bacteriocidins or antimicrobial proteins. So the microbiota is very important in the defense against C. diff. And if you look for the modified microbiota modifying therapy, there are several options, isn't it? So we did have the probiotics, the prebiotics, the symbiotics, the fecal microbiota transplantation, and the bacterial mixtures. Fortunately, we do not believe in the probiotics, the prebiotics, and the symbiotics. There are insufficient evidence nowadays to think that those factors can have an influence on the clinical course of CDI or on the prevention of recurrences. The question if they can prevent the development of CDI infection or colonization has not been resolved yet. So therefore in the Netherlands we do have the fecal microbiota transplantation as a treatment option for patients with multiple recurrent CDI. And Lister here is the head of that National Donor Fetus Bank, and we treat on an annual basis about 60 to 70 patients nowadays with multiple recurrent CDI. And we are indeed, just as Benjamin, also convinced that in the near future we will switch from fecal microbiota transplantation to live biotherapeutic products in the form of bacterial mixtures. So the take-home messages are: one, indeed, the epidemiology of C. diff is still changing, and we do see new hypervirulent strains from unexpected sources sometimes, and those hypervirulent strains can not be recognized by conventional diagnostic methods and conventional typing techniques. We really need the whole genome sequencing. Second is that CDI is also an important disease in the community. And finally, the concept of altering our gut community by microbial intervention is very useful for the prevention of recurrent CDI. So we think that this was the 2021 update of the treatment guidance document for CDI in adults, written by Geoffrey from Plan, in which we have that microbiota intervention already mentioned and advised for the treatment of multi-recurrent CDI, but that will probably be increased in the coming years with live biotherapeutic products or with other interventions of the microbiota. So I thank you for your attention.
Hey, thank you very much, Ed, for this presentation. Yeah, thank you. I was going to ask Benjamin to join us with the video. Thank you, messieurs, for this wonderful presentation. I have seen a few questions pass by. They are not necessarily retained over time on the script, but Garinia Suneja was asking concerning valerate, whether it was acting on Clostridium difficile, as you show, Benjamin, but also on other microbes, whether it has been tested on other microbes.
Yes, in that set of experiments, we showed that for a number of ribotypes of C. difficile, including 027, there was a sort of dose-response relationship whereby the higher the dose of valerate, the less the vegetative growth of C. difficile. However, when valerate at different doses was incubated with a number of commensal bacteria, it didn't seem to have very much effect on their growth, except on one of the Bacteroides members, I think uniformis, at very high doses.
Okay.
But otherwise, very, very little. Yeah.
And valerate in this context is produced by bacteria? It
is, but we don't, it's not very well characterized.
I was going to say, do we know who's doing the job, actually?
Yeah, exactly. The question we're always asked, and we don't have a clear handle on which bacteria are producing.
Okay.
Progress, yeah.
You mentioned the connection with the tyrosine conversion to paracresol. I know paracresol is incriminated in autism. Is there any connection with the carriage of C. difficile and autism, for example?
Very interesting question. I don't know offhand.
Not seen any of that. Yeah. Okay. I'll continue with you in series. Actually, Ed, don't worry. I'll get to you in a minute. You mentioned the work by Stefan Schreiber in Kiel. Filtered feces acting as well as a complete material, and so obviously that opens the possibility of many functionalities being still acting. What's your vision on that? Do you think that one metabolic mechanism could be able to recapitulate the full effect, or will it require a combination?
Yes, when I hear people ask this debate, I hear two ways. I hear on the one hand that regulators would like it much more if we can just simplify it to a few functions that are well defined that we can treat in a pharmacological way.
Sure, one molecule.
On the other hand, I hear this is far too reductionist. These are complex ecological systems, and you're never going to be able to reduce it to a very small number of functions. These all act in tandem. So I think the jury is still out on that at the moment. Yeah.
Okay, I'll have a question, I guess, to both. I mean, FMT for recurrent Clostridium difficile infection is let me put it this way: it's not very demanding in that you can use a donor stool and put it with a saline and mulch it with aerobic conditions and filter, and it will still do the job, basically. In other clinical conditions, we are putting extreme precautions to maintain the diversity of the microbiota. So what's your feel on that? Let's start with Ben and then translate or transfer to Ed.
Do you mean what's my view in general of use in
the fact that the technicalities, the process for preparing the drug with a donor stool is not very demanding. You can do it aerobic and it will still work. You can just mash and filter and it will work. So, I mean, it will probably perturb quite a bit even the metabolites themselves.
Yes. Yeah, I don't know. You mean if we've got something that works, you mean why look for something else? Why not just stick with what we've got?
Well, I was just wondering whether you have an idea of why this is so easy to do in a way.
Yeah, I don't know. I suppose it's the unique degree of disruption of microbiome composition and functionality of that company's CDI in particular.
In the context of CDI.
Yeah, that just anything you throw at it, however you select your donor or however you prepare it, is so easy. It's obviously very unique in that aspect, as we've seen by comparison for other FMT studies in non-CDI conditions. IBD being an obvious example where there's a clear rationale for why you might expect benefit, but there is obviously something very unique about the degree of disruption in CDI.
Ed, what's your message on that?
Yeah, I agree. I think that there are not many requirements to the FMT and to the donors of FMT for the treatment of patients with recurrent CDI. I mean, it's probably what is so interesting that there is also an immediate effect. So if you do have patients with, let's say, refractory CDI or severe CDI, if you treat those patients with FMT, that has been done in a few occasions, you see a rapid effect with resolution of the inflammation and then disappearance of the symptoms. So I don't know if it's only an effect on the microbiota. I think there's also something on as an immunological effect that influences the inflammation and can be used as an, let's say, as a kind of a model for the treatment of severe CDI infections. I think that the studies of Benjamin with the metabolites and with the role of valurate, they are very interesting. So far as I know, there are some effects of, let's say, of bacterial communities that are isolated, cultured, encapsulated and given to patients, but I'm not so impressive on the effects of those cultured bacteria and live bacteria products.
Yeah.
And so that is probably not an answer to the question. It's not a specific group of bacteria that do have characteristics like bile acid hydrolysis. No, that's not. I think it are other other properties such as the production of certain metabolites or small chain fatty acids. So
yeah. There might be more than we know in terms of also compatibility between donor and recipient. And so just in terms of ecology, that might count a lot. And of course, in terms of metabolites, that might matter quite a bit. One question I wanted to raise for you, Ed, is you have several examples where you, I will say, confirm the potential for asymptomatic carriage. When you say the work of Quinten showing that there are 43 negative and 41 asymptomatic positive, that's 50%. I don't know whether it's a random pick that led you to that observation, but what's your perception? Is there something like a prevalent carriage, non-symptomatic carriage of Clostridium difficile in the community?
Well, if you define asymptomatic carriership as patients who do have C. diff in the intestinal tract, spores of C. diff or vegetative cells of C. diff, temporarily or continuously, but without symptoms, then it's probably a good definition. But if you look to the microbiota composition, they are not, let's say, healthy. I mean, they do have a disturbance of the microbiota. So that brought us to the hypothesis that it's not just a normal inhabitant of the intestinal tract. Okay. It's a microbiological symptom of a disturbed microbiome.
Okay.
That's in contrast with everything that has been reported until so far about, let's say, the high frequency rate of asymptomatic colonized children. But also in those children, you can see that the microbiota in children is different than the microbiota in adults.
So then when you say that Bacteroides prevalence is associated with a higher risk, and Fusicatinibacter is a candidate for protection. Well, for this last one, have you been looking into in vitro demonstration of antimicrobial potential against Clostridium difficile?
Yeah, we have selected a few more bacteria. We've selected now 3 or 4 bacterial species. And some of those species are also present in products of, let's say, Vedanta, and we tested those strains in vitro on the growth of C. diff and the production of toxins by C. diff. We can clearly see some interaction between those strains and the C. diff. So yes, there is some interaction that certain bacteria can produce compounds that inhibits the growth of C. diff, and that can also, let's say, lead to a rapid sporulation, or that can inhibit the growth, can also inhibit the toxin production, and can decrease the toxin concentrations in the intestinal lumen. So there is an interaction on several levels, but still it's, let's say, it's an in vitro model in a cultured medium with 2 or 3 bacterial species only. And that may be, again, completely different from the situation where you do have a whole intestinal microbiota with more than 2,000–3,000 bacterial species and archaea and fungi present. So it's promising, of course, but it's probably not the only factor.
Okay, thank you very much. There would be many other questions on my part, and also we had one request from one of the attendees, but I think that we are getting to the closure of the session. So let me just give just a few words. I will start thanking you, messieurs, very, very much for the presentations that were really fantastic, fascinating, very informative, and really top up to date. We thank Ferring Pharmaceuticals for supporting the webinar series, and I just want to invite our attendees to not miss the next session that will be on June 28th, so coming month, and that will be on clinical picture and testing of CDI. So once again, thank you very, very much, and we'll close the session here. I will pass on to both of you the message that was sent just before we closed, so that we may see whether you can address that for the attendee that sent it, Dr. Kristen. Thank you very much.